Welcome to our presentation on the cell cycle and mitosis.
A key stage of interphase is DNA replication. DNA is a form of nucleic acid that holds the genetic material of the cell. Another form of nucleic acid is RNA. Nucleic acids are composed of nucleotide chains, which is made up of three components: pentose sugar, in DNA it's deoxyribose, phosphate group, which is joined to the deoxyribose sugar to form the DNA backbone, and nitrogenous bases. In DNA, they are adenine, guanine, cytosine, and thymine. The nucleotide components join together to form two polynucleotide strands which coil around each other to form the double helix structure. The sugar phosphate chains that make up the two backbones of the helix have opposing directionality. The two polynucleotide strands network through hydrogen bonds between the bases, forming base pairs. This means adenine complements thymine and cytosine complements guanine. It is the order of the base sequences that determine how proteins are coded. RNA only has a singular polynucleotide chain, a sugar phosphate backbone containing ribose. In addition, the nitrogenous base thymine is replaced with uracil. A section of DNA is referred to as a gene, and certain genes are expressed to generate specific proteins. Proteins are important for cell growth and repair, replication of DNA, and overall cell regulation. They are also required for a process known as apoptosis, when a cell is damaged beyond repair. During interphase of the cell, protein synthesis takes place, which involves both DNA and RNA. RNA carries express genes in the form of messenger RNA to the ribosomes in the cytoplasm, whereby ribosomal RNA and transfer RNA decodes the expressed gene and converts it into proteins. Protein synthesis includes two steps: transcription, which takes place within the nucleus of the cell, and translation, which occurs in the cytoplasm outside of the nucleus.
Gene regulation controls gene expression, which is essential for how proteins are synthesized. There are different protein mechanisms that increase or decrease the production of genes. There are two types of regulators involved. Positive regulators are involved in gene expression and are proteins which allow the cells to keep dividing because there aren't any faults. Positive regulators are affected by cues in their environment. Proteins involved in positive regulation include CDK, cyclin-dependent kinase, an enzyme protein, and also cyclin. Negative regulators are involved in gene repression and are proteins that stop cells from dividing due to possible faults. Negative regulators are also affected by cues in their environment. Proteins involved in negative regulation include P53, which initiates apoptosis if the cell is damaged beyond repair.
The cell cycle refers to the period in which cells grow before proceeding a regulated cycle of division, which in this case is mitosis. However, not all cells undergo mitosis. For example, red blood cells and neurons do not divide. The cell cycle can be defined in three key stages, which are interphase, mitosis, and cytokinesis. There are three processes in interphase that cells must pass in order to successfully achieve cell growth. There are G1 phase, S phase, and G2 phase. Not all cells will enter the G1 phase. Some will leave the cell and not actively grow or divide. This phase is G0 and is known as the quiescent state. G1 is the first stage of interphase in a eukaryotic cell. In this phase of the cell, protein synthesis occurs and the cells grow in preparation for the following stages. The cell doubles its number of organelles so that during mitosis, the two new daughter cells have identical components. Chromatins are present and are packed together in a nucleus to avoid any damage to their DNA. This helps control DNA replication and gene expression. There are also centrosomes present within the cell. There are a few checkpoints during the cell cycle in order to regulate the process and avoid mutations. G1 checkpoint is the first, and this checks that the cell is at an appropriate size. It also checks that growth factors such as nutrients, temperature, and room for growth are all suitable. These growth factors will help to stimulate cell division, and it also checks for any DNA damage. If there are any damages to the components, the cell will undergo apoptosis.
The S phase only begins when the cell has grown enough to contain double the DNA. During the S phase, DNA is replicated. Although the number of chromosomes after this process has doubled, we still count them as 46, but as 46 chromatids. We do this because we count the number of centromeres, which is the area that joins the sister chromatid. During the S phase, the protein P16 is present. This protein helps to prevent some cancers by suppressing tumours. The G2 phase is the final part of interphase. This is the second growing phase where the cell continues to grow and produce new proteins. This phase helps to prepare the cell for division. By this phase, two centrosomes have formed which contain two centrioles. These centrosomes help to organize the microtubules in the cell. Centrioles help to develop spindle fibers during mitosis. The second checkpoint of the cell cycle is the G2 checkpoint. This checks for any DNA damage and ensures the DNA has replicated fully.
Mitosis is the stage in which nuclear division occurs. It takes place after the interphase period. The first stage in cell division, DNA and proteins condense. Prophase is split into two sections, early prophase and late prophase. During early prophase, the chromatin fibers condense up and the nucleus begins to vanish. In the final stage, late prophase, two sister chromatids, the two identical copies of each chromosome, are attached via the centromere. The centrioles are at opposite ends of the cell. The protein spindle fibers appear and attach to the centromeres of each chromosome. The nucleoli disappears and the nuclear membrane breaks down. During metaphase, the chromosomes line up at the equator and join the mitotic spindle. Long protein filaments, called the kinetochore microtubules, pull the sister chromatids back and forth until they align at the equator. After metaphase, there is a metaphase checkpoint. This ensures the cell is ready to divide. The control point checks the chromosomes have attached to the spindle fibers correctly and they align properly at the equator. If the cell passes these checks, it moves on to the next stage of cell division. During anaphase, the centromere divides, so the sister chromatids can part and become distributed to the opposite poles of the cell. A protein called cohesin bounds the sister chromatids together, but during anaphase, this protein is removed so that the sister chromatids can part. Microtubules attached to the chromatids shorten so they are pulled towards the centrosome. This stage happens after the cell has passed the spindle fiber checkpoint.
Telophase is where the nuclear membrane reforms, chromosomes uncoil, the nucleoli reappears and the spindle disappears. This is the final stage of mitosis before cytokinesis. At this stage, the sister chromatids reach opposite poles and the chromosomes are grouped to each end and become surrounded by small nuclear vesicles that are in the cell as it reforms. As it reforms, two nuclei are created in one cell. During this stage, the dissolution of the kinetochore microtubule occurs. The chromosomes are now decondensed. Cytokinesis is the process in which the cytoplasm and organelles divide. The actin and myosin filaments contract to separate the parent cell into two identical diploid daughter cells. Each daughter cell contains a diploid chromosomal set that is identical to the parent cell. Each new cell has a plasma membrane forming around it.
Cancer is a disease which is caused by a growth defect of cells. This means cells will divide rapidly and uncontrolled during mitosis due to a mutation in a single cell. Uncontrolled mitosis can be a result of alterations in genes, including tumor suppressant genes like p53 and oncogene caused by the mutant cells. Therefore, if these negative regulators are faulty, it will then not be able to suppress these cancerous cells and initiate apoptosis.
Thank you for watching our presentation.